Hey Saves, I'm just going to talk you through these. You'll have to apologise. No, I have to apologise that it's just on my email screen like this. It's too early in the morning for me to figure out how to get them off my email. Um, so my favourite topic we're looking at, and as I said, um, I've been teaching this to my year 12, so it is very fresh in my mind. So we've got the famous diagram here showing crossing over. Okay, and then the production of the recombinants. So the word recombinant means genetically shuffled. So basically, when crossing over occurs here, the new chromosome or the new chromatid on that side has been genetically shuffled. It's got a new genetic code. So we've gone from um, recessive A, recessive B, recessive C to recessive A, recessive B, dominant C. Okay, so a new order of alleles, genetic reshuffle, that's the recombinant. Non-recombinant are the ones, the parent chromosomes that haven't, their initial allele sequence. But remembering, they're actually recombinant from their parents. So that's how this whole idea of variation between generations occurs. Now, let's have a look at our questions. So first up, define the term recombination. So that's a genetic reshuffle of alleles, okay, compared to the parent chromosome. The reshuffle occurs during crossing over. And the phase that occurs, prophase, I believe it is. I will double check that in a second. I can never remember all of my phases off by heart. It's tricky. My students have taken just to, they have their mnemonic, PMAT or whatever it is, and then just by each of them, they just write a bullet point of the key thing that occurs in each phase. Discuss the difference between a recombinant chromosome and a parental chromosome. So the parent chromosome has got the original gene sequence or allele sequence, the original allele sequence for their generation. Okay. Crossing over occurs, it reshuffles that allele sequence to create the recombinant chromosome. So the recombinant chromosome is the chromosome with the reshuffled allele sequence. I'm just repeating myself because if you hear it repeated, if you don't get it once, it may drop, like the penny might drop afterwards as such. Now your answers? Yep, so that's cool. Just get the word allele sequences into there. So remembering alleles are just different types of genes. So when you look at this here, you've got an allele for a dominant A and a dominant A. Okay, whereas once the crossing over has occurred, and this could happen in the parent generation as well, you've got a dominant C, for example, and a recessive C. So the recessive C and the dominant C, they're still the same trait, but one's recessive, one's dominant. So it might be like having a gene for you yourself could have a gene for blonde hair, because there is blonde hair in your family, and a gene for black hair. Blonde hair or brown hair. Blonde hair is obviously recessive because you have brown hair, but you could carry both genes. So because the genes are different but for the same feature, they're called alleles. All right, that's that one. All right, I need to get to the next. It's up there. Alrighty, based on the information provided, identify the allele sequence in each of the parent chromosomes. So I'd write parent equals, and then you've got a dominant A, dominant B, dominant C for parent 1, and recessive A, recessive B, recessive C for parent 2. Then you would have recombinants, and recombinant recombinant 1, hang on, just let me go back to that question. Yeah, so you just need to identify that these parents are homozygous, okay, because they've got the same allele, oops, sorry about that, the same allele on both chromatid. Then identify that the recombinants are heterozygous. So we've got a recessive A, dominant A, dominant A, dominant B, and then we've got a dominant C and a recessive C. So I would say recombinant chromosome 1, chromatid A, or no, sorry, try not to use A because you're using that in your genes. So chromosome 1, you could say chromatid 1, A, B, C dominant, chromatid 2, A, B, recessive C, recombinant chromosome 2, chromatid 1, recessive A, recessive B, recessive C, dominant C, that is dominant, isn't it? My eyesight's bad, and chromatid 2, recessive A, recessive B, recessive C. So basically what that question is showing, that you can see the difference between these being homozygous, so the same gene, same allele, crossing over has occurred, and we've now got two heterozygous recombinants. And it's asking for the gene. It's a tricky question because it says, based on the information provided, identify the allele sequence. Yes, it's easy in the parents because they're homozygous, so you've got the same gene, but it's same allele. It's just tricky when it comes to recombinant because you've got to write it for each chromatid as such. Let me know if that one doesn't make sense. I may have just talked in circles and confused you. All right, meiosis is responsible for the production of gametes. Discuss the significance of crossing over in terms of offspring developing a unique genetic identity, and we call this variation. So crossing over increases genetic variation, okay, which means each gamete has a different arrangement of alleles. So it increases genetic variation. That's our answer there. Identify another event. Random assortment. So I'm going to pause this. I've got a slide to show you. Hang on. Okay, my favourite slide to answer this question. So our question was, identify another event that occurs during meiosis that enables the offspring to develop a unique genetic identity. So there's three processes, right? Let me find my PowerPoint again. The first one is crossing over. The next one is the formation of the gamete. Okay, so when the chromatids split and form the gametes, you have a different combination of chromatids. So remember, chromatids being one arm of the chromosome. You have different combinations of chromatids in each of the gametes that are formed. So we call that random orientation. Okay, so that formation of all these gametes with the different genetic material is random orientation. And then after that, the fusion of gametes then also promotes genetic variation. So you've got crossing over, then you've got random orientation where they have different combinations in each gamete. And then we have this random fertilization where, you know, one sperm and one ovum with all these different genetic variation within them then fuse. Okay, so in answer to your question, I would go with that one, that random orientation of chromatids, but you could also choose actual fertilization, okay, and the fact that the two gametes are unique in their genetic code. which then would roll on to this one if you did random fertilization, because you've now got discuss the biological advantage of sexual reproduction with specific reference to genetic diversity. Because of crossing over, random orientation, and random fertilization, you have 3 key points of ensuring genetic variation. If you have an organism that reproduces asexually, they are a complete clone of the parent. Their genetic code is identical. Okay, so to increase genetic diversity, we need that genetic variation, because one generation is always different to the generation before. And then the next generation that comes from the new generation will again undergo those 3 different methods of genetic variation. Okay, I will leave our video there. Flick me through any more questions you've got.